Transcription factor binding site prediction. Aims and objectives for this lecture then are to refresh some of the basics of transcription initiation in eukaryotes, to introduce transcription factors from a bioinformatics perspective. At the end of the lecture you'll be able to describe the problems of predicting where transcription factors bind. You'll be able to summarise some experimental methods for identifying transcription factor binding sites. You'll be able to show how transcription factor binding sites can be encoded. You'll understand the problems of finding transcription factor binding sites in the genome and be able to give an overview of how we approach this. And finally, you'll gain a feeling for the performance of these methods. Why do we need to predict transcription factor binding sites? Well, they are key elements in the regulation of gene expression. So they give us a general understanding of how gene expression works. They can help with gene finding, as was discussed in the previous lecture. And mutations in transcription factor binding sites can lead to disease. Experimentally validated sites are limited in number, and most experimental methods have relatively poor resolution, and thus one needs to find the actual site within the experimentally identified site. So let's start with some background. In the last lecture we saw the typical eukaryotic gene structure consisting of a promoter, an open reading frame composed of exons and introns, and we talked about promoters being nucleotide sequences to which RNA polymerase binds to initiate transcription. In addition to that, we have these transcription factor binding sites that help with the polymerase binding. So in prokaryotes, RNA polymerase has strong affinity for the promoter, and that means that the basal transcription rate is high. In eukaryotes, however, RNA polymerase 2 and 3 pre-initiation complexes don't assemble very efficiently. So the basal transcription rate is low, and other proteins, these transcription factors, are needed for effective initiation. So transcription factors are defined as sequence-specific DNA binding factors. They're proteins that activate the initiation of eukaryotic transcription. They may be constitutive, which means that they will work for many different genes and they don't respond to external signals. Or they may be regulatory. These work with a limited number of genes and do respond to external signals. Some recognize upstream promoter elements. These influence the initiation at the promoter to which they are attached. And others recognize targets within enhancer regions, and these influence several genes at once. So the traditional view is that transcription factors activate formation of pre-initiation complexes by either making direct contact with polymerase 2 or making indirect contact, so the transcription factor binds to the DNA, that interacts with another protein, which in turn interacts with RNA polymerase 2, or they can introduce some sort of DNA bend. However, we now know that some can modify histone proteins, affecting nucleosome positioning. Some bend the DNA into a specific shape, bringing other transcription factors into contact with the pre-initiation complex. And some do not bind DNA at all, but form protein-protein contacts with the pre-initiation complex. Now, I apologise that the scanning of this picture is not very straight, but here we just see a few examples of transcription factors and their binding sites taken from the 1999 edition of the Brown Genomes book. We now believe that there are as many as 2,600 different human transcription factors. So what can we do about prediction? Now, when we look at these, we have a consensus sequence, but of course it is only a consensus. That doesn't mean that we always have exact matches to these sequences. Some of the sequence patterns indeed are incredibly small, so GATA1 binds to the sequence GATA. Now, in the modern databases of transcription factors, the shortest binding site is 5 base pairs long, which is the transcription factor called PAX4. The largest is 30, and most are in the range of 9 to 15, with a mean of 12.2 base pairs and a standard deviation of 3.7. If we took that example of GATA, so just using 4 bases, if we assume that all 4 bases are used at the same frequency, then the probability of seeing this sequence GATA is 1 divided by 4 to the power of 4. In other words, 1 over 256, which is 0.0039. Now, given that we have around about 3.2 billion base pairs in DNA, that means that by multiplying that number by the probability, we get the expectation value, the number of times we expect to see this pattern if everything is completely random. So we would expect to see it 12.5 million times in the human genome. Now, clearly that cannot be a real signal of information about what the GATA1 transcription factor is doing. So we need to take a more complex approach to prediction. First of all, we need some way of encoding the patterns that describe the binding site in a more informative way than just looking at, for example, GATA. And then we need some method to scan these patterns against the DNA, and it needs to be more intelligent than just simple naive scanning of the form that I've just described. But before we go into the details of that, let's have a look at experimental methods for identifying transcription factor binding sites. Now, the first of these is called the Electro-Mobility Shift Assay, or EMSA. Now, this is an in vitro method. It isn't used actually in cells, so we're working with DNA and the transcription factor proteins in a test tube. Now, what we can do is realise that if we split the DNA into fragments, then the mobility of the DNA is going to be different in a mobility assay when DNA is bound to a protein. So essentially what we're doing is running a control, in this case, of DNA only, down a gel, and here we're running the DNA and a protein that doesn't bind to it, and here we're running the DNA with a protein that does bind, and we see that the band is shifted. The second type of assay is also done in vitro, and it combines DNase-1 cleavage with electrophoresis. And what happens here is that when a protein is bound to the DNA, it shields the DNA from being cleaved. So what we have here on this gel is a control band here. We have different concentrations of protein here. So if we have a lower concentration of protein having an effect, it's going to indicate strong binding. So what we see here is that there's a whole set of bands that are missing compared with the controls at either end where we don't have the protein present. And we see that here as well. And we see it across all the different concentrations of protein, indicating that this is a strong binding site. In this case, we have some loss of bands, this one in particular here, at the higher concentrations of protein. So this indicates a weak binding site. A third in vitro method is known as systematic evolution of ligands by exponential enrichment, or SELEX. Now what this does is it starts with a large library of DNA fragments, and basically what we're doing then is selecting for fragments that bind to our protein of interest. So we're using affinity chromatography. We have the protein anchored on a column. We run our DNA library down the column. We wash off the non-binders and elute the binders. Alternatively, rather than using affinity chromatography, we can use EMSA that we described earlier. So this is illustrated in this cycle here. We're starting by generating lots of different DNA fragments. We select and we then extract those with the required property, in other words, binding to the protein. And we can amplify those, and we can then go through mutation cycles. So the amplification step then will typically be by PCR, and potentially during the replication we can use low stringency copying to allow these mutations to accumulate. Subsequent rounds then use higher stringency elution, and at the end of this we can sequence the best binders. A modification to SELEX, again an in vitro technique, is SELEX-seq, and this is exactly the same as the SELEX method, but in SELEX you're somewhat limited by the number of these oligos that you can end up sequencing, usually a maximum of about 100. In the case of SELEX-seq, we use next generation sequencing, and that allows us to characterize more than 10 million oligos. In addition, SELEX requires many selection rounds, whereas SELEX-seq, because we're able to do more sequencing, allows us to use only 1 or 2 selection rounds. Now, those techniques that I've described so far are all in vitro techniques. What about in vivo? Well, the first of these is known as chromatin immunoprecipitation, or CHIP. And what we do here is we cross-link DNA with proteins with which it's associated. So we add some sort of cross-linking agent into the cell. The DNA protein complexes are then sheared into fragments of around 500 base pairs. And the cross-linked DNA protein complexes are immunoprecipitated with a protein-specific antibody. Finally, the DNA fragments can be purified and sequenced. There's also a variation of this known as chip-chip, which is doing this chromatin immunoprecipitation on a chip. So the first stages are similar, that we're cross-linking and shearing and then selecting with a specific antibody, but we can then release the DNA and label it with a fluorescent tag. We can then hybridize the fluorescently labelled DNA with a microarray of fixed DNA sequences from the genomic region of interest. And then we can identify the matches from fluorescence and perform computational analysis to find out what we have. ChIP-seq is another variation of CHIP, which is done in vivo, but uses next-generation sequencing. So this is a bit like SELEX-seq compared with SELEX in the in vitro context. So basically all we do is the chip type experiment, but we can sequence everything, and a single sequencing run can scan for genome-wide associations with fairly high resolution of 100 to 300 base pairs. However, some degree of computational statistical analysis is required, so-called peak calling, in order to find regions of the genome that are enriched with aligned reads. Another variation on ChIP-seq is known as ChIP-EXO, and this is kind of the latest standard in this kind of approach. So this starts with the kind of ChIP experiment, but then uses exonucleases to trim the protein-bound DNA. And they have claimed single base resolution of the binding site. So this compares with finding a region of between 100 and 300 base pairs with ChIP-seq. Here we're finding just the actual section that the protein binds to, the transcription factor binds to. So let's compare the performance of these methods. So EMSA and DNase 1 footprinting are generally not that good. They can identify non-specific binding sites. All the methods really tend to find regions that are larger than the actual binding site, with the possible exception of ChIP-EXO. Now if we look at these graphs on the right, these are two different regions, and what we've got in grey is the result of doing ChIP-ChIP. So in other words, these are the regions that are identified as binding to something. And what you can see is that while there is a peak here, it really is spread out and we're getting signals all over the place. And the same thing happens in this example. Now with ChIP-seq we're getting a much better, clearer signal here, and indeed here as well. But as you can see from the scale here, this section is around about 100 base pairs. This one's a little bit narrower, maybe 75, 80 base pairs. But then when we do the ChIP-EXO, we're finding a very much smaller, sharper peak, indicating exactly where the transcription factor binds. Nonetheless, ChIP-seq is largely the method that most people are still using at the moment. Now, as I said earlier, a transcription factor binding site is typically between 9 and 15 base pairs long, and it's degenerate. In other words, it's not an exact pattern of bases. There's some variation. And all the methods, with the possible exception of ChIP-EXO, tend to produce wider areas, especially ChIP-ChIP and ChIP-seq. So we need to identify where the actual transcription factor binding site is within that predicted region. And various motif discovery methods have been employed to do this. Now don't worry too much about the detail of how these work, but basically they fall into two categories. One is enumerative methods, and these basically just try to identify overrepresented strings of characters. We can examine the frequencies of all DNA strings from these wider areas, and from that we can look for patterns that are overrepresented. The advantage of this as a technique is that there is less chance of getting stuck in local optima. We're more likely to find the correct sequence overall. The second approach is to use what is known as a probabilistic method. These generate a local multiple sequence alignment and learn various descriptive parameters. These fall into different categories. Again, don't worry about what these actually mean, but they fall into expectation maximization, Gibbs sampling using Bayesian Monte Carlo, and so-called greedy approaches. The advantage of this type of method is that we can deal better with arbitrary motif model variations. For example, motifs that vary in length, whereas we can't do that with the enumerative methods where we're just looking for frequencies of strings because we have to look at a fixed length. Just to point out again then that the reason that we're trying to do this is we're doing experimental work to find where a transcription factor binds. But in most cases, the region that we identify from using these methods identifies a much longer region of DNA than where the actual transcription factor binding site is. So all of these sequences that we get will have the transcription factor binding site in them, but that may just be, say, 10 bases in a chunk of 100 bases or more. So what we need to do is use these sorts of methods to home in on where the actual binding site is. Now, MEME is one of the most popular methods for finding these patterns. However, it struggles with huge data sets from genome-wide techniques. So, for example, ChIP-seq can produce more than 10,000 sequences per run, and MEME is not very good with these sorts of numbers. So we can use a random subset of the sequences, but this may end up being inaccurate. So there are newer tools that have been designed for dealing with these huge datasets, including Chipmunk, Homer, ARGADM, and MEME-CHIP, which is a variation of the MEME method. And a recent evaluation from a few years ago suggests that ARGADM generates the best motifs from ChIP-seq data. Now, a useful side effect of these methods that are able to find a motif within a set of sequences is that those motifs can then be described in a way that can be used for finding transcription factor binding sites by just looking at a single piece of sequence instead of having to align a whole set once again. At this point, we're going to take a break, and in the next part we will focus on the prediction of transcription factor binding sites rather than the experimental analysis of where they are.